Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before the starting the topic, please like and subscribe the channel. In the last session, we studied about lysosomes and peroxisomes. In today's session, we study mitochondria. In this diagram, we can see inner membrane, outer membrane, cristae, and matrix of mitochondria. Mitochondria are spherical or oval or rod-like bodies about 0.5 to 1 micrometer in diameter and 7 micrometer in length. Erythrocytes do not contain mitochondria. The tail of spermatozoa is fully packed with mitochondria. Mitochondria are the powerhouse of the cell, where the energy released from the oxidation of foodstuffs is trapped as chemical energy from ATP. Mitochondria have two membranes. The inner membrane convolutes to form folds or cristae. The inner mitochondrial membrane contains the enzymes for electron transport chain. The fluid matrix contains the enzymes for citric acid cycle, urea cycle, and heme synthesis. In this diagram, we can see that cytochrome P450 is heme synthesis. Mitochondria also contain specific DNA. The integral inner membrane proteins are made up of mitochondrial protein synthesizing machinery. However, the majority of proteins, especially the outer membrane proteins, are synthesized under the control of cellular DNA. In this diagram, we can see mitochondrial DNA, which is a part of mitochondria. The division of mitochondria is under the command of mitochondrial DNA. Mitochondrial ribosomes are different from cellular ribosomes. Antibiotics inhibiting bacterial proteins synthesis do not affect the cellular processes, but they do inhibit mitochondrial protein synthesis. In this diagram, we can see ribosomes, which is a part of mitochondria. Taking into account these evidences, it is assumed that mitochondria are a parasite which entered into the cell at the time when multicellular organisms were being evolved. These parasites involved provided energy in the large quantities, giving an evolutionary advantage to the cell. The cell gave protection to these parasites. The perfect symbiosis in turn evolved into a cellular organelle called mitochondria. In this diagram, we can see ATPase enzyme. The smaller portion of ATPase enzyme is F0 and the larger portion is called F1 portion. Before moving further, I want to tell you that please like, share and subscribe the channel. Your one click on the subscribe button shall motivate me to prepare further videos. Also, any suggestions can be given in the commentary box. So moving to the further, it is plasma membrane. The plasma membrane separates the cell from external environment. It has a highly selective permeability properties so that entry and exit of a compound are regulated. The cell metabolism is in turn influenced and probably regulated by the membranes. The membrane is metabolically very active. The enzyme nucleotide phosphatase, or also known as 5' nucleotidase and alkaline phosphatase, are seen on the outer part of cell membrane. They are therefore called ectoenzymes. Membranes are mainly made up of lipids, proteins, and small amounts of carbohydrates. The content of these compounds vary according to the nature of the membrane. The carbohydrates present as glycoprotein and glycolipids. Phospholipids are the most common lipids present and they are amphipathic in nature. Cell, cell membrane also contains cholesterol. In this diagram, we can see the plasma membrane. Within the phospholipid bilayer, we can see glycoprotein and glycolipid. Fluid mosaic model theory of plasma membrane. The lipid bilayer was originally proposed by Davison and Daniel in the year 1935. Later, the structure of the biomembrane was described as fluid mosaic model by Singer and Nicholson in 1972. In this diagram, we can see that within the two layers of phospholipid membrane, there is extracellular fluid, carbohydrate, then transmembrane protein, glycoprotein, glycolipid, peripheral protein, cholesterol, etc. Phospholipids are arranged in bilayered with polar hair head groups oriented towards the extracellular side and the cytoplasmic side with the hydrogenic core. The distribution of phospholipid is such that choline containing phospholipids are mainly in the external layer, is in those containing ethanolamine and serine containing phospholipids in the inner layer. Each layer is 25 angstrom units with the head portion equal to 10 angstrom units and tail portion 15 angstrom units thick. The total thickness is up to 50 to 80 angstrom units. The lipid bilayer shows free lateral movement of the components hence the membrane is said to be fluid nature. However, fluidity enables the membrane to perform exocytosis and endocytosis. However, the components do not freely move from the inner to outer membrane, also called flip movement, or from outer to inner membrane, also called as flop movement. According to apoptosis, this is that is programmed cell death, flop movement occurs. In this diagram, we can see flippases and floppases. Flippase shifts inward, whereas floppase shifts outward. The flip-flop movement is catalyzed by enzyme. Flippase catalyzes the transfer of amino phospholipids across the cell membrane. Floppase catalyzes the outward directed movement which is ATP dependent. This is mainly in the role of ABC proteins, that is ATP binding cassette proteins, mediating the efflux of cholesterol and extrusion of drugs from the cells. The multi-drug resistance associated with P glycoproteins is a floppase. The cholesterol content of the membrane alters the fluidity of the membrane. When the cholesterol content is increased, the membrane becomes less fluid on the outer surface but more fluid in the hydrophobic core. The effect of cholesterol on membrane fluidity is different at different temperatures. At temperature below Tm, cholesterol increases fluidity and thereby increasing membrane permeability, whereas at the temperature above the Tm, the cholesterol decreases permeability. In this diagram, we can see that graphical representation of cholesterol effect on temperature. With cholesterol, it is the straight line, whereas it is a sigmoid shaped curve without cholesterol. Here ends our this session. Kindly like, share and subscribe our channel. Thank you.